So hello, I'm Yannis Nevers for a postdoc at the Decimo Lab at the University of Lausanne, and I'm going to present you OMArk, a tool we developed for quality assessment of protein coding gene repertoires. So what I mean by coding gene repertoires, first definition, is a set of coding genes annotated on a given genome sequence, so for a given species. In public database, they are often available as proteomes, where you have one protein sequence by genes. These data sets, like all, are subject to quality issues, and in this case, they can be missing genes, fragmented genes, inclusion of non-coding regions in the repertoire, or contaminations. And sadly, we lack a tool to detect all of these issues at once. That's why we developed OMArk. So OMArk rely on the OMA database, which is an orthology database with 2,500 species across the tree of life. And for it to run, OMArk will need a small representation of this database that's called OMAmer database. Then you just need to give it your proteome that you want to assess. And the first step of the workflow will be to place all of these protein into gene families. For that, we use OMAmer, a tool we developed in the lab to do fast camera-based mapping of proteins within gene families. So the output will look like that. Each protein sequence will be attributed to a gene subfamilies. And that's all you need to run then OMArk that will give you a quality assessment of the proteome. So now I'm going to give you the principle behind OMArk. So first you have a proteome, and the thing OMArk will need is to have for this proteome an ancestral lineage. So that means the latest ancestral clades that contain the species of these proteomes and that have at least 5 representative in OMA. OMArk will normally automatically select it either from the user provided tax ID or from the placement from OMAmer if not. And once you have the ancestral lineage, OMArk will select conserved ancestral genes for this lineage. So there are genes families that were defined at this ancestral level, and so that kind of correspond to the ancestral gene repertoire, and that are still present in at least 80% of the species. Using that, and because we expect them to be present in most representative of the lineage, we can use that to get a completeness assessment of the set. So genes of the ancestral genes that are present in the proteome or that have homologs in the proteomes will be considered as present, either a single copy or duplicated, and the ones that do not are considered as missing, and this is kind of how we measure incompleteness. This may sound familiar to you because it's really similar to what other tools like BUSCO uh, hack to measure completeness too, although here there's a few methodological difference. Another thing that's more original to OMArk is that then it uses a more global set of genes that are all gene families that are at least one representative of the selected lineage. We use that to get an assessment of the quality of the whole proteome. So the idea is we expect most genes in the proteome to actually have homologs in close species. These genes that map more closely to gene families to this lineage, we will call them consistent. The ones that do not, either they do not map to any gene families, we can't say anything about them, we call them unknown. If they map to other families that come from the other species all across the tree of life, here we call them inconsistent. So it doesn't kind of make sense for this proteome. Of course, if OMArk notice that a lot of these inconsistencies always go to the same lineage, here it's more probably contamination and it will be reported as such. So this is the idea of the whole repertoire assessment. Now, here it's more, I will go with a more practical example. We have the big-eared turtle proteome. OMArk will attribute the clade Archelosaurus with 10,000 conserved hogs. On these hogs, we see 8% are missing. That kind of means the proteome is slightly incomplete, but otherwise still close to 100% complete. So it's still okay. Then we pass to the whole proteome assessments. We have 20,000 genes. 90% of them are consistent. It makes sense and it's quite encouraging. And we see very few of them are either inconsistent or unknown. But we do notice there is a bit of contamination here that HOMArk attribute to actinobacteria, probably meaning that the genome assembly integrates some contaminant sequence. Another thing OMArk does, finally, is also report information about the gene structure. Like this black bar here correspond to partial mapping. That means only part of the sequence have homologs in the gene families, and that part of it is not has no homology at all. So it's kind of inconsistent gene structure here. The other part of things we report is fragments. That means when we have a protein that map to a gene families, but it's much smaller in size than the representative from it. And you can see here, there's quite a few of these, probably meaning there's kind of still work to do to improve that. Now, let's go to practical application. Why would you want to use HOMArk? So let's say you have a particular clade that interests you and you want to select a few proteomes to run your analysis. Here we have an example of the concentric proteomes. We run HOMArk over all of them and we would want to select the best quality one. So from the cursor I glance over here, you can also already see these three that are nearly complete. So that's kind of the one you will want to look at. And if you look at the whole proteome assessment, they also are the ones with the less proportion of inconsistent genes, partial mapping of fragments. So you'll likely probably want to select one of these, maybe this one because it looks the best of the three. Another thing for which you can use HOMArk is to compare two annotations for the same species. That's what we do it for the honeybee, an annotation from July 2020 on Ensemble and the current one. What we can see by comparing them side by side is there has been a noticeable improvement in terms of the whole proteome, like there's much less fragment, partial mapping, or inconsistent genes. That's quite encouraging, even though apparently it comes at the cost to a bit more missing genes. So here, to conclude, so I hope it convinced you to give OMArk a try. We hope to publish it quite soon, but in the meantime, you can already try it. It's available publicly on GitHub as a Python package on PyPy, and you can also use it without installing anything on a public web server on this link or using this QR code. So now I'd like to thank you for your attention and to thank the lab for their support during this project, especially these people that contributed directly. And I'll be open to question. Thanks.